Okay, so this is the protein part of the lecture. So proteins have the abbreviation capital P, capital R, capital O, and that's just by convenience. There's really no reason for that. The monomers for proteins are amino acids. Amino acids are abbreviated capital A, lowercase a. There are 26 amino acids in nature. But humans only use 20 to make their proteins. To make a protein, you put amino acids together. So you have like this generic amino acid 1 bonded to an amino acid 2. The bond between amino acids is called a peptide bond. Okay. So the bond between amino acids is called a peptide bond. That's also why proteins are sometimes called polypeptides. Examples of proteins, and there's millions of different proteins, but examples are collagen. And collagen is a protein that's used in our body to create structures. And collagen is very strong. It's in our skin and our hair and our nails. It's in our bones. Tendons have a lot of collagen in it to keep them strong. And then another example is lactase, and lactase is a type of enzyme. So enzymes are made of proteins. Okay. This protein over here in this picture, this is just here to show you what a protein would look like. You don't have to memorize any part of that. Proteins have 4 different conformations. And those conformations are like arrangements, or how the proteins are put together. All proteins have a primary conformation. So the 1 with a degree symbol here, that just means primary. And the 2 with a degree symbol means secondary. So the primary conformation is simply the sequence of amino acids. So you have amino acid 1, peptide bonded to amino acid 2, which is bonded to amino acid 3, to amino acid to the nth, because there are many proteins have thousands and thousands of amino acids. Some only have a few amino acids. But this protein, the primary sequence for proteins, is just simply the sequence of amino acids. The secondary conformation is either an alpha helix or a beta sheet. Beta sheets are also called beta pleated sheets. Okay, or means the same thing. An alpha helix is simply a spiral of proteins. The beta pleated sheet looks like a piece of folded paper. Okay. So this could be a secondary, or the secondary possibilities are either an alpha helix or a beta pleated sheet. Not all proteins have alpha helices or beta pleated sheets, though. Tertiary structure is how the protein is folded. So the tertiary structure is how the protein is folded. And the tertiary structure is really important for enzymes, because if an enzyme isn't folded correctly, then it won't work. The quaternary structure is the number of proteins in the molecule. So for example, hemoglobin has 4 proteins in it. Hemoglobin is a molecule that's inside our red blood cells that carries gases. Okay, so these pictures show those examples, or it shows examples of those 4 protein conformations. So the primary structure is here. Each of these, this is the primary, each of those circles represents 1 amino acid. And so this amino acid 1, 2, 3, 4, 5, 6, so on and so forth. So the primary structure is simply the sequence of amino acids. Which amino acid do you have first? Here are examples of the secondary structure. So you've got a beta-pleated sheet here, and then the alpha helix there. Over here we have the tertiary structure. And in the tertiary structure, you can see that the protein, which was that primary structure, is now folded with its alpha helix and its beta-pleated sheet. So there's the alpha helix, there's the beta-pleated sheet. Okay, so the tertiary structure, it's all folded up. And the quaternary structure is this, and you see we have this one lightly shaded protein and a second darkly shaded protein. So that's showing that this quaternary structure for this protein has 2 protein molecules in it. This next one shows the same thing, just a different way to look at it. So with this one, we have our primary structure, which is just a sequence of amino acids. So you have amino acid 1 and 2. And we have the secondary structure showing the beta-pleated sheet, or the alpha helix. And in the tertiary structure, you can see that that beta-pleated sheet here, or the alpha helix there, is showing up in how the protein is folded, because this whole protein is folded a certain way. And then the quaternary structure is like this. Here's that hemoglobin molecule we talked about that has 4 different proteins in it.